Hello everyone. In this presentation, we will explore the use of deep learning in drug discovery, particularly in drug-target interaction prediction. And we will discuss the challenges posed by limited training data and introduce the concept of transfer learning as a potential solution. Let's start by understanding the steps for the drug discovery. Drug discovery is defined as the process of identifying the roles of bioactive compounds to develop new drugs, and it is usually one of the initial steps in a drug development pipeline. Traditionally, screening is accomplished via low or high throughput experimental bioassays and then continue with lead optimization. Since this whole process is time consuming and costly, machine learning methods have been proposed to speed up the first couple of steps. Here we have some challenges. Limited bioactivity data for protein raises concerns of overfitting in high-dimensional input features, and deep learning models tend to memorize without enough training data. Here, if you look at the chemical space, we can see that there are more than 1 billion compounds, and nearly 12,000 of them are FDA-approved or experimental drugs, which indicates that a vast amount of them awaits to be explored. And for the protein space, number of human proteins are around 20,000, and nearly 80,000 of them have 3D structures, and around 2,700 of them are known drug targets, which presents an exciting opportunity for developing new therapies. The process of drug discovery can be divided into three categories, which are prediction of drug-target interaction, compound-protein affinity, and compound properties. Highly successful results were obtained with deep learning models compared to the traditional methods. However, the practical impact of these deep learning models in drug discovery has unfortunately been limited, since it requires large amounts of data. The number of bioactivities for target proteins over the data in Campbell version 29 can be seen in this chart. Only 6% of the proteins have many bioactivities, which is more than 1,000. On the other hand, a large number of proteins have few bioactivities. Almost 64 of them has 100 or lower bioactivities, and which is very hard to train. And that is one of the reasons why we have chosen this subject to study. Here, our approach is to first train a deep neural network with the source training dataset of large size, and then to reuse this pre-trained neural network as an initial configuration for fine-tuning purposes with a small size dataset, and also train the model from scratch using the original network and using a support vector machine. And finally, using the target test sets, perform many numbers of experiments and obtain performance evaluation and comparison results. Now I will continue with methods. In traditional machine learning methods, learning is performed without considering past learned knowledge in other tasks. Therefore, knowledge is not retained or accumulated from one task to another task. On the contrary, in transfer learning, a source dataset and a basic network on the source task are trained first. The learned attributes are then reused or transferred to a second target network to be trained on a target dataset and task. With this way, learning process can be faster, more accurate, and need less training data. When the target dataset is significantly smaller than the source dataset, transfer learning can be a powerful tool. Our method consists of two stages. In the first stage, we build a feed-forward neural network model by training with the source protein family with many bioactive compounds, and we call it source model. In the second stage, for a target protein family with low number of bioactive compounds, which is called target model, we have three modes in this stage. The first mode is full fine-tuning. The weights of the pre-trained source model are used as initial values for the target model. In mode 2, which is called feature transformer, One of the hidden layers of the pre-trained source model responsible for feature extraction is frozen, and only the remaining layers are modified using the target training data. And mode 3 is shallow classifier. The output layer of the source model is replaced with a shallow classifier, and the feature vectors extracted by the frozen bottom layers are used as input to the classifier for training with the target data. Here we use support vector machine, random forest and gradient boosting machines as a shallow classifier. And for the task we use binary classification. Here we are trying to predict for the given compound whether it is active or inactive for the target protein or protein family. And for the featurization, we use small strings and convert them into ChemProp features, which are 300-dimensional feature vectors. And ChemProp tool uses a message passing neural network to construct these input features. Now I will continue how we construct the datasets. To generate our training and test datasets, we utilize the Campbell database version 29, which has more than 18 million bioactivities. The datasets underwent a rigorous filtering process to ensure the quality and relevance of the data. After applying these initial filtering steps, we remove duplicate bioactivities in the final steps, and then entries which has a p-Campbell value greater than 7 are selected as active entries, and the entries which has a p-Campbell value less than 7 are selected as inactive entries. Additionally, compounds were converted into ECCB for fingerprints and clustered to prevent bias towards specific chemical series during training and evaluation. To construct the datasets, we have chosen 6 protein families, which are GPCRs, ion channel, kinase, nuclear receptor, protease, and transporter. After applying the filtering steps that I mentioned in previous slide, we obtain this table. This table provides us with an overview of the dataset statistics. And here we have selected nuclear receptor and transporter as a target dataset since they have the least data. And to evaluate the performance with lower numbers of drug-target interaction data points, we created 8 smaller and balanced target training datasets, and their sizes are between 2 to 4,000. To construct the training datasets, we used 5/6 of the original dataset, and the remaining 1/6 of the dataset used for tests. And for the evaluation metrics, we used log area under curve, F1 score, and MCC. And in the remaining slides, we used MCC as an evaluation metric to show the results. And finally, I will show the results. Here is the performance results for transporter dataset where the source model is kinase. When the training dataset size is less than 100, transfer learning outperformed training from scratch, including the reference and the base models. As the target training dataset size exceeds 100, the performance of transfer learning approaches become comparable to the training from scratch. However, the transfer learning still has the advantage of requiring fewer training epochs, which reduce training time significantly. And in this slide, the results for transporter, where all other protein families are used as a source model, are given. And we can see that all families gave similar performance in overall, but if we use kinase as a source dataset, we get the best performance increase in transfer learning. Here you can see a loss graph for one of the small datasets. And when transfer learning is used, target model starts from lower loss values compared to the reference model. Therefore, a lower number of epochs are generally sufficient for training, which significantly reduce the training time. Here, the source models are directly used for the test of the target data. Although these results are still acceptable, it is easy to observe that even a very small amount of data, such as 2 target samples of training via transfer learning, boosts the performance. And we also perform similar tests this time for a single protein. Here we have selected AKT1, and AKT is a critical kinase involved in regulating cellular processes related to the cell growth, survival, and metabolism. And its activation and downstream signaling are tightly regulated and play important roles in both normal physiology and disease. And AKT1 is annotated with EC number 27111. Here you can see the 27111 and its parents and what they represent. And we also apply some filtering steps for AKT1 which are similar to the previously explained filtering steps. But this time we used 4.3 p-Campbell value as an active threshold, which is around 50 macromolar. And for inactive compounds, we have selected compounds which are interacting with transferases proteins. And after applying the filtering steps, we obtain this table. Here we use 27111 as a source dataset, and then AKT1 is used as a target dataset. And we perform 1,000 different experiments, and here 50 active and 50 inactive compounds are randomly selected from the original AKT1 dataset. And after applying those experiments, we obtained the best model for AKT1. Here you can see the precision recall curve for the best model for AKT1. And for this model, we obtained 0.84 area under precision recall curve score, and we obtained 0.76 F1 score. And for this F1 score, the threshold is around 0.85. And we perform additional tests for AKT1 using the best model that I mentioned in previous slide. In this case, the compounds that are known active or inactive for AKT1 are tested. And we made sure that these compounds are not available in the training set. For inactive compounds, all our 3 methods gave very small prediction score, and for active compound, transfer learning methods gave very high prediction score. And as a discussion, when transfer learning is used, it's significantly give better performance than the training from scratch, especially if the dataset size is less than 100. Learning with minimal data is possible with as low as 2 compounds, and other transfer learning techniques can be also effective, such as feature transformer and shallow classifier. And deep transfer learning effective even with enough data, here faster convergence compared to the training from scratch. And no training and short target specific fine-tuning are also possible. Here source model can be directly used for inference without using any training data, and target specific fine-tuning, for example with only 2 target samples, boosts the performance. For the future work, we are planning to screen the drug bank dataset for AKT1, and we are planning to test possible candidate drugs for AKT1 via wet lab experiments. We are also planning to develop additional models for more single target proteins such as CDK1 and CDK2. And we are planning to build a new transformer model for compound representation, which contains millions of compounds, and we are planning to use graph attention network. Here finally we have a web-based server, and it is called tl4dti.council.org. And the first, it consists of 3 models, and the first model is the fine-tune model, which allows the user to create a target model from a pre-trained source model via transfer learning, and then this model can be downloaded using the interface. The second model is the predict model, which lets the user perform prediction directly using one of the pre-trained source models. Here the default source model is kinase, and then the predictions can be downloaded using the interface. And the third model is fine-tune and predict model, which is the combination of the previous 2 models. Here you can all do these operations without running any code, by just using only the interface. And we have also a GitHub page where you do these operations without any dataset size limitation.